Afternoon. Welcome to the annual Listinsky lecture. I'm really delighted to see this standing room only crowd. But for those of you who actually want to sit down, I'm happy to host you across the hall in the pathology chair's office. We have a conference room and we can put it on Zoom in there if you can't find a seat. So in addition to welcoming everybody here, let me tell you a little bit about the Listinskys. Jay and Kathy Listinsky were faculty members for many years here. Kathy was a surgical pathologist, and Jay was a breast mammographer. But Jay had a secret passion, and that was he wanted to do basic research. And it's very hard as a radiologist, or for that matter, a diagnostic pathologist, to do research without a lab and supplies and technical support, et cetera, et cetera, et cetera. So we reached out and allowed him to have that within the pathology department. And he was particularly interested in glycobiology, especially fucose, and how it was tied into neoplasia. And for many years, he worked successfully there, wrote a bunch of papers, and had a hell of a good time. Well, eventually, he and Kathy moved off to the Midwest to a different institution. And about 10 years ago or so, he unfortunately developed malignant neoplasm. And before they died, he wanted to thank us for supporting his work, and thus the Listinsky Lecture. That has allowed us to bring to campus once a year essentially the foremost glycobiologists in the world, and this year is no exception. I should say, as I wrap up, Jay only demanded one thing in the commitment letter for the lectureship, and that was that donuts would be served for the residents. So you'll see in the back, there are donuts as promised. To introduce our speakers, Dr. McConaughey. Thank you all for coming again.
Thank you, Dr. Siegel. I heard that he's under the weather. He still came. We really appreciate that. Welcome to 8th Annual Listinsky Lecture Series. First of all, before I say I'm not going to talk anything about the series, but I would like to thank all my pathology family, including the faculty, our trainee, staff, and particularly our organizing people. This Fatima, she has put a lot of time on that, organizing this wonderful seminar. We should all, thank her for that one. Now, I have to do a very difficult job because I need to introduce someone who is doing this glycobiology for 15 years. And I will not take more than a couple minutes because you want to hear him, not me, right? So I'll be brief. So today we have, it is my privilege to introduce Dr. Schnaar from Johns Hopkins. As you know, he's like a extreme researcher and a scholar, and he's professor in the pharmacology and neuroscience, and he's also interim director of department and pharmacology and molecular science at Johns Hopkins. So one thing you will see in his CV, those has seen, so he very much stay in that Baltimore area. He just did his undergraduate from University of Michigan, then all his Ph.D. Then briefly he went to NIH, just other campus in Baltimore. Then came back again, did complete his postdoc at Johns Hopkins and never ever moved from there. So always he has been there in Johns Hopkins. So for the 50 years, he has embarked on like remarkably career that you can talk about. I don't have too many words to introduce him, what he has contributed in the field of glycobiology, not just basic mechanism, how this can be used for a therapeutic purpose. As of written in the CV, he has 237 publications, maybe more. Maybe in next few days, he have a couple more articles, already accepted. So he has many book chapter, and some of them are used in the field, not just science. He has a wonderful, like career for like training next generation scientists, postdoc, graduate student, all that one. If you look at his recognition, if you ask me any NIH institution he not funded, there is none. All of them are. He got funding from all. He has like 68 grants all for his career. Maybe I'm missing someone. I'm just talking about some NIH grant. So then he has served many leadership roles. He was like president of glycobiology, and he has done on many NIH study section, you can name it. So without further delay, it's my extreme pleasure to welcome our speaker, and good to have our privilege to have a speaker like that for this special glycobiologist. Please welcome me to join him, like for the seminar series.
Thanks all for coming. I really appreciate it. And I want to thank Dr. Siegel and Alkani, who are involved in bringing me here. It's been a really nice couple of days. I've had some great meetings, trainees, a lot of fun talking to the trainees, those that are in the field, those who are out of the field. And I want to thank Fatima, she here? Yes, in the back of the room, Igdura, who has been tireless in arranging my coming here. Thanks a lot, Fatima. I want to start by saying, I know not everybody is familiar with the field. This is something that is evolving. Talking about glycobiology, many people think back on carbohydrate chemistry if they ever had it. And this is a very different field than that. So I've put together an introduction, glycobiology, a perspective in the field. And as you heard, 50 years in the field. And I think I can present kind of a perspective that I hope will give you an appreciation for why I spent so long in the field, and maybe for how this might impact your thinking about the biomedical discovery universe. And also to honor Dr. Listinsky, who had a particular interest in fucose. So I'll throw that in as well. Only after I've done that, and I think primarily that's the message that I want to share with those of you who might not have been invested in the field, to give you a perspective on what this is about from my 50 years. And then to move on to some of our more recent work, which has to do with discovering some of the molecules that are involved in immune regulation. So my disclaimer, recently I was recruited to be part of the scientific advisory board of a company that's focused on Sialics as immune modulators. So take that as you will. So let's get on with this. Even today, this, and yeah, I realize I'm on, figure out how to mouse over this. Hang on. Even today, this is the picture that one gets of the cell surface. You can look it up on Google, so it must be right. It is not, by the way, this was published when I was a senior in my undergraduate education. I remember the publication in Science we're also excited about. And it's based in part on this view, which you can find in cross sections of EMs of any cell. And what you'll see here is that you have this bilayer that is distinguishable. Its thickness is the thickness of a lipid artificial bilayer. And so we know that's exactly what it looks like. This happened to be stained with a hydrophobic dye, which invested itself into the lipid portion of the membrane. And it missed something. When you use a more hydrophilic dye, ruthenium red, you get this. Let me blow this up so you can see this. The lipid bilayer, which everybody that reads Google knows is the surface of the cell, is covered with something that never stains. And one of my students was talking about this at a presentation. In fact, the member said, what kind of cell is that? And the answer is it doesn't matter. In fact, every eukaryotic cell is covered with a dense and complex array of glycans. And in fact, evolution has failed to generate a single living cell, prokaryote or eukaryote, devoid of surface glycosylate. So the picture you get from Wiki is that the cell surface is kind of a red rock desert with some cactus on it and some yucca trees, and those are the glycans. But in fact, that's not at all. So I turn to a quote from Marcus Aurelius, not to be outrageous, but I enjoyed this quote because it kind of fits science for a lot of us. And it's this thing, and we're talking about this thing, if you know what's there. What is it in itself, in its own constitution? What is its substance and material? What is its form? And what's it doing in the world? And that's what I've spent 50 years devoting myself to. So the cell surface is not a red rock desert. It's a forest canopy. If you look at the mass of the molecules on the cell surface, it's kind of like the trunks of a tree. You say, well, that represents 90% of the mass. That must be about what it is. But clearly, if you're not seeing the leaves, you're missing the whole point of the forest. This impacts a lot of functions. I had a real enjoyable breakfast with trainees this morning, and one of them astutely asked, so glycans, are they more the biophysical? You know, is it the hydrated structural molecules, or are they informational? And the answer is yes. Both are true. They're very much involved, and evolution has taken advantage of them in both these areas. So going back to Marcus Aurelius, what is the constitution of this? And we pretty much know the constitution of this. And before I go on with this, I will say that a mistake we have made, testing. Let me use this. A mistake we've made in the field over the years is to send out the signal that this is so complicated. Glycans fit together and they branch, and there's so many of them. You can have different ones at different places. And some of my colleagues have even written reviews that said, we'll never know all the structures. That's wrong. This is a paper I wrote for the Journal of Leukocyte Biology in 2015 called Glycobiology Simplified. Why do I call it Glycobiology Simplified? Because in fact, if we all decide here just for today, just to focus on humans, rather than all the other microbes and lower parasites, et cetera, which do incredibly clever things with their glycans. If you look up monosaccharide, there's a database with 700 of them. Okay, I'm not working in this field. There's too many building units. If we focus on humans, there is nine building blocks. You can get this. And they are joined together. Use this. They are joined. So here are the nine. Many of them are just very simple. I'll show you one in a minute. Very simple tweaks on glucose. And they fit together in very well-defined ways. Why? Because it turns out that these are not template-driven. They're enzyme-driven. And there's about 200 10 glycosyltransferases that build these. We know what they are, and we know what they build. And this is a kind of theme and variation. You have kind of a base unit of glycans on N-linked glycoproteins or O-linked glycoproteins, to which these terminal structures can be placed. And these terminal structures can drive molecular recognition, which is one of the functions that I'll show you is important in human physiology and pathology. And these are glycoproteins, glycolipids, and proteoglycans, all of which, by the way, in a way, it's Coles Newcastle. There are people here that are more astute about these than I am in some of these that have been working here at UAB, which historically has also been a great center for glycobiology research. These then are manufactured by each cell type and distributed on their cell surfaces. I'll exclude some others that I'm not going to talk about that aren't on the cell surface. Most are, like you saw, and form a repertoire that is the face of the cell that identifies the surface for molecular recognition. Let me give you one example. If you were a pathogen and you were coming down to land on that forest canopy, would you bind to the trunks of the trees? Of course not. You'd bind to the leaves, right? So the outermost leaf is sialic acid. Influenza virus, I hope you all got your vaccine, recognizes sialic acid on the surface of your airways. That's how it gets in. Why? Because that's the easiest way to get in. That's what's out there. It didn't look it up on Wikipedia. It's got to deal with what's really out there. So we don't express these in order to become targets of pathogens. We use them for our own molecular recognition. Molecular recognition is only one, as I said in talking about that astute student question, is only one of the functions of glycans, but it's one I've devoted my career to. And there's lots of interesting targets that are physiologically and pathologically important in that field. And that's what I'm going to be talking to you about today. The way we think about this is that we have lectins, glycan binding proteins on a cell that can then recognize a glycan, one of these arrays, typically by its outermost portion, either on another cell to mediate cell-cell recognition, or in the extracellular milieu, as you'll see when I get talking about our own work, to regulate immune cells and other cells in the body. And we talk about these binding proteins having evolved a carbohydrate recognition domain. I'll show you that as an example. And we talk about this grouping of glycans as being the carbohydrate binding determinant that's written. Now, as I mentioned, this is all about molecules that spread out in space. They're hydrophilic, and they have little tweaks. And you may have studied in school glycans as straight lines with hydroxyls going this way and that way. No glycobiologist thinks of sugars because they are whoops. That was 2 hits, not 1. Now I have to come over and change this, sorry, to a, if I can get to it. I'm sorry, that's the thing is kind of not behaving itself. If you'll just give me a second, let me get out of this and fix this. So I can show you what I want to express on this. There we go. There we go. I'll do it this way. That's fine. So in the You want to know something? I'm going to tell you about this. And I'll stay at the microphone. So it turns out that these circular sugars, all of them have a hydrophobic center, believe it or not, surrounded by hydroxyl groups that are just sticking out. And in glucose, for example, if you look at either side, both sides are hydrophobic and so can sandwich into a hydrophobic pocket. And then you have all these hydroxyls that can make hydrogen bonds. And if you look as simply swapping one hydroxyl from a comfortable equatorial position to an axial position, suddenly one side of it has a big hydrophobic hydrogen bonding group on it. That means it won't fit into the same binding site. And the reason I'm so interested in sialic acid, and I will go on to sialic acid, is that in particular, and I don't know why this is doing this, but in particular, sialic acid is a bit more complex. It's got a carboxylic acid, you'll see that can bind to things. It's got a glycerol side chain with 3 hydroxyls hanging around, and it's got a hydrophobic side, all of which can get involved. So let's get out of that, which was messing us up, and look at what recognizes these. In humans, there's about a dozen families, 4 of which are shown here, of glycan binding proteins or lectins. These, each one has a story. Each one is involved in a physiological and or pathological process that we can intervene in. And for this introduction, because it involves fucose and a specific, well-documented example of essential physiological function, I'm going to talk about the selectins. And I'm going to start, if I can get over to it, by talking about inflammation. You know this as well as I do. So what we have here is a mesenteric vessel going through a thin tissue in a rat. And the investigator, Michael Lawrence, has put a needle nearby and is releasing lipopolysaccharides to imitate a bacterial infection. And when he does that, the neutrophils, which were just flowing by, crowd around that position against the wall, the endothelial lining of that vessel. And why? That's how you fight bacterial infections, but they can't do it from there. First of all, thank goodness, neutrophils flow around and don't get into your tissues because they're kind of nasty. But when you need them, you need them. And after 15 minutes, they begin to flatten out and move in. And by an hour later, those neutrophils have spread out into the tissue where the bacteria are to kill them. And that's inflammation. You have some pain, swelling, redness, et cetera, and even as you saw at the very end there, the next door neighbor vessel bed does not have that happening because it's so carefully controlled. This starts with glycan recognition. It starts by glycan recognition, and the situation is that you have a glycan on the surface of the neutrophil. It's always there. It's special and it's ready. When you get a bacterial infection nearby, binding proteins that recognize that pop out. Some are prepackaged in the endothelial cell and pop out quickly. Others are synthesized and are responsible for a longer term response. Then and only then can you engage other cell adhesion molecules and eventually move through the endothelial into the tissue. This is what we know. There are 2 of these glycan binding proteins that are involved. They're called P-selectin and E-selectin, and they have these glycan recognition domains out there at the end where they're going to pick that up. Thank you, Dr. Listinsky, if you're interested in fucose. Fucose is one of the key parts of, you can see 4 different sugars in a particular grouping that are involved here, and I'll show you why that's so important in just a moment. And down here, you can actually see the protein amino acids involved in that recognition. There's bound calcium and amino acids that make coordination and hydrogen bonds to the fucose, which is really important. The galactose with its hydroxyls in a particular place, and the sialic acid with its carboxylate. All of those are required for this to happen. How do we know it's important? Well, if you knock out so this is a bacterial clearance assay in a mouse model. Mouse clear bacteria pretty well. And if you knock out E or P-selectin, they're a little less efficient at that. But if you knock both of them out together, you cannot clear bacteria. And we know this in humans because of individuals like this rare case of what's called LAD2, leukocyte adhesion deficiency 2. Now, unfortunately, this child has some developmental issues that we're not going to talk about today, but that was not the child's major problem in the clinic. The problem was that he could not fight bacterial infections. And many of you know that the typical white cell count is about 5,000 per microliter in normal humans. And when he was born, he had exactly that. But within days, his neutrophil count in the blood shot up tenfold. But they couldn't, these are white cells, leukocytes, neutrophils, I'll use those interchangeably. They couldn't get to where the infection was occurring. Each one of these exacerbations over a year's time were hospitalizations for this infant, constant antibiotic treatment, failure to thrive. Then somebody realized that the child had a strange blood group type. You could say, what? Turns out you and I have O or A or B blood group type. And it's because blood groups types are driven by glycans. If you have a fucose alone on a galactose, you're O. If you have an extra N-acetylgalactosamine, you're an A, and extra galactose, you're a B. What if you don't have any fucose? Well, that's a strange blood group type. It's called the Bombay phenotype. And investigators realized, as I just showed you, that fucose is absolutely required for binding to selectins. And they said, well, wait a minute. We know the enzymes that put that fucose on for blood group isn't the same enzyme as puts the fucose on for the Sialic X selectin ligand. So it's not an enzyme that's involved. What if it's metabolism of fucose? So they fed this child fucose on this day during an exacerbation. Within days of starting fucose therapy, neutrophil levels returned to the normal range. There were no further infections, and antibiotic prophylaxis was discontinued. This is an example that drives home that cells, in this case, communicate by glycans and glycan binding proteins to drive essential functions. And if we understand this, it provides opportunities. That's another quote we use in the pharmacology department. If this is true, what does it imply? It implies that there could be therapeutics based on this. And there haven't been dramatic therapeutics yet, but my friend John Magnani at Glycomimetics did develop this molecule that has a whole fucose on it and some galactose, sialic acid-like parts, and some mimetic parts. And he was able to show that in an acute inflammation model in humans in phase 2 trials, which was inflammation due to vaso-occlusive crisis, in sickle cell disease, which involves neutrophil binding, that he could, and this is measuring the amount of pain reliever self-administered by the people in the hospital to show how acute this inflammatory response was. And in red, you can see when they were treated with this drug, they did not have this inflammatory response. So we can understand glycans not as straight lines with hydroxyls pointing in other directions, or cacti and yucca plants, or as the coating on a lovely looking frosted donut, thank you, but as essential molecules underlying molecular and cellular communication. And if I leave you with anything, that's what I want to leave you with. If you run into this in your work, and you may, please reach out to glycobiologists. We're a very open community. We want to learn about new ways that these are involved. So in the last half hour of this talk, I'll talk about two vignettes that we've worked on. Both involve another side of inflammation, and both involve molecules called Sialics. And they are modulators of immune cell function. So let's talk about those. So I'll show you this in more detail in a moment, but so-called I-type lectins, of which Sialics belong, have immunoglobulin-like domains anywhere from 2 to 17, which out at their furthest end have a glycan binding protein that binds sialic acid. So these are sialic acid binding immunoglobulin-like lectins discovered over the last 20 years or so. And there are 14 of them in humans, yes, numbered up to 16 because 2 of them turned out to be pseudogenes. They were discovered by searching the databases. And almost all of them, all but one, are expressed on overlapping, as you'll see in a moment, sets of immune cells. And many of them have these intracellular so-called ITIM domains that are immune inhibitory and change the process of immune. So we're going to talk about Sialic 8, a little less so Sialic 9. And you can see they're about the same size, and they have similar structures, but they have different distributions, different functions, and we'll talk about Sialic 8 in particular. So here's a list, not to be taken, I know the writing's kind of small, but just to show that each of these Sialics is in different overlapping subpopulations. And you'll see that Sialic 8 happens to be on eosinophils, basophils, mast cells, which are allergic inflammatory cells, and microglia, which we realized only a couple of years ago, and I'll get into that if I get to it before the hour's up. Sialic 9, which is very similar in structure, is on neutrophils, monocytes, B cells, and T cells, but mostly neutrophils. Both of these have the same structural domains. Both of them are immune inhibitory. The Sialic family developed to take advantage of all the different ways sialic acid can be presented on the cell surface. Not to make it overcomplicated, there's 4 ways it can be. It can be on the cell surface molecularly, and it can be on those are the outermost structures, and it can be on then internal structures that vary as well to give it specificity. So here are the 4 ways. Doesn't really matter, but I know that Susan Bellis and her lab is working on ST6-gal1 that makes this structure, which is particularly overexpressed in cancers. It can be attached to 2,6 to GalNAc, which is the T antigen that is a cancer target. It can be attached to itself in 2,8 linkages. And there's the Sialics recognize differentially all of them. So 1 binds the 2,3, 1 binds the 2,6, some bind the 2,8. So it's evolution has developed these different similarly structured molecules on different immune cells to be able to engage different sialic acids in their environment for different outcomes in the immune system. We're going to talk about Sialic 8, which, as you'll see, recognizes a linkage that has an extra sulfate on it, and Sialic 9, which overlaps but in a different structure, and I'll get to that. So what are the cornerstone observations? I'm going to go to the first one. What are the cornerstone observations that are important to us as pharmacologists or physicians or biomedical researchers in Sialic 8? It's expressed on allergic inflammatory cells, particularly eosinophils and mast cells. Cross-linking Sialic 8, and this was done artificially using an anti-Sialic 8 antibody, results in eosinophil apoptotic death and blocks mast cell release. You think, well, that sounds like a good idea. If I have an acute allergic reaction and I want to kill the eosinophils and stop the mast cells, and that hasn't gone beyond the recognition of Alicos, a company spun off by a colleague of mine, Bruce Bachner, which humanized this anti-Sialic 8 antibody to use in allergic inflammatory diseases. So this is mediated, interestingly, this inhibition is increased when the cells are activated. You can kill more eosinophils if they're activated. So to us, the message we're getting on this is the downslope side of an inflammatory event. Build up your inflammatory cells in response, but then turn them off. When they're activated, they become sensitive, then you kill them or turn them off. So this is a model by Jackie Schaefer, Bruce Bachner's spouse, who happens to be an artist, like a chemical artist. The concept is it comes in that these glycans, what are these glycans? That's what I'm going to talk about, come in, engage these Sialics, cross-link them, and then signal, in this case, for eosinophilic death. So there's opportunities. This is an axis of control, a natural axis of control for which there are potential therapeutic opportunities. Here are the data from Bruce's lab showing this. So on the left side, you can see that if you have unactivated, no IL-5, human eosinophils in vitro, over 24 hours, these are tough experiments to do, about 10% of them apoptose, and you can increase them to about 20%, so you get some increase. If you activate them with IL-5, they're healthier in vitro, and you kill more of them by cross-linking their Sialic 8. And if you go right into the lung tissue and flush a late phase allergic response patient and get those eosinophils and treat them with this cross-linking antibody 2E2, almost all of them can be killed over a dose response with 2E2. So this is the basis. This was the basis for spinning off Alicos, which is using this. Mast cells, same kind of thing. If you activate a mast cell with anti-Fc epsilon cross-linking, these cells release histamine. If you treat them with by cross-linking their Sialic 8, you can decrease that. They release prostaglandins. Likewise, you cross-link their Sialic 8. It's inhibitory. You're turning off your mast cells. You're turning off your eosinophils. Same thing, flip side with anti-Sialic 9 for neutrophils. Here are neutrophils that, as you can see, von Guten et al. Did this work, different group. If you have human neutrophils in vitro and you activate them with GM-CSF and cross-link them with Sialic 9, then you can apoptose neutrophils. So these are immune regulatory molecules on this, glycan binding molecules on the surface of eosinophils and neutrophils. And this spreads to other cells as well. So here we have our model. We have Sialics on the surface of the immune cell, and then somewhere out in their environment, you have some glycan. What is the glycan? That's our job. We focused on asking what in the natural environment of the eosinophil and mast cell during disease is engaging it. I will say that evolution has moved very rapidly on glycans because they're also used by pathogens. Influenza virus is a good example. And we have moved pretty far away from mice, which makes some experiments difficult. So all the work I'm going to be talking to you about is in humans and human tissues. We have made some mice that are expressing human genes, but I'm not going to get into that. And mice have Sialics themselves. They're just different and in some cases recognize different things. So now we're talking about having a carbohydrate determinant, and we want to know what that carbohydrate determinant is. So how does the field do this? Well, it's been estimated that with the glycosyltransferases that are available in humans, you might be able to make 4,000, 3,000 different glycan structures that would fit in a glycan binding site. So far, we have synthesized, not we personally, but the field has synthesized maybe 1,000 of those. And different groups have put them down by stamping them in microarrays so that you can overlay and ask, what does my molecule bind to? What does Sialic 8 bind to of all the possible structures? So in this case, we had 600 structures on an array, and we overlaid it with Sialic 8. And it picked out 2 of those. And they had almost the same structure. And you can see the structure to the right has a sialic acid bound to a galactose, which also has a sulfate. Pretty unusual. Interestingly, Sialic 9 did not bind to either of these if you go vertically, but it bound to something real similar that had the sulfate 1 sugar over. It's like, wow. If you had an enzyme that could put the sugar on the galactose, you make something that can kill eosinophils and turn off mast cells. You have another enzyme that puts it on the N-acetylglucosamine, you can kill neutrophils. This is an exquisite system to build signaling molecules for defined outcomes in controlling inflammation. We know now why that is, because Propster published the NMR structure of Sialic 8. And what we find there is it's got a really nice pocket where a arginine makes a salt bridge to the sialic acid carboxylate, but it's also got another pocket down here, where you have positively charged groups loving the sulfate. So evolution has built this to hug this molecule. Sialic 9 won't bind to it. It's this level of specificity of glycan structure. And if you look at binding affinities, what you find, and this kind of struck us, and Propster did this, that if you don't have sulfates, or if you have the sulfate on over here on the N-acetylglucosamine, you have really poor binding. But if you have the sulfate on the galactose, or both of these sites sulfated, you're great. And suddenly struck us both of those sulfated, because there is a molecule, hello, sophisticated proteoglycan aficionados in the audience, that has this structure. It's called keratan sulfate. It doesn't matter, but we make something that looks like this. And keratan sulfate is a repeating 2 sugars in which every glucnat gets sulfate. Well, that doesn't help us at all. But after you make this, and these can be long, you can add a sialic acid at the end, and another enzyme can add this sulfate. So you can make this structure by taking 2 specific enzymes. We know the enzyme, a sialyltransferase and a sulfotransferase, that build this at the time of need to be able to engage Sialic 8. So this is our synthetic ligand for Sialic 8. You can see that binds great. This is the synthetic ligand for Sialic 9. And just this month, somebody was talking about Hert-Jan. Hert-Jan Boons at the CCRC published that he had built artificially this set of structures to test this, where he put the sulfate here or here on both. And I won't go into all the details, but every structure that had the sulfate on the galactose, these 1, 2, 3, 4 structures bound Sialic 8 great. They didn't bind Sialic 9. As a matter of fact, even if you had both the sulfates like this structure, Sialic 9 wouldn't bind to it because it is distinguishing whether well, down here, it's distinguishing whether the glucanac by itself is sulfated. So all of this subtlety and recognition is to drive biology. I'm not going to take you to the end of the whole story, but I am going to show you what it is that binds to this in vivo in humans. And we now have this molecule in hand and can work with it to look at its effects. That's what we do. To do this, we had to start with human airways. And we're looking at upper air. Well, we looked at all airways, but we'll look particularly at upper airways where allergic inflammation targets in asthma and nasal passages. And in the upper airways, you have an epithelial layer, you have a submucosal layer with submucosal glands, and you have cartilage. So in allergic inflammation, you're targeting this. I'm not going to go into COPD, so I'll skip that for time. So what we did is we took Sialic 8 itself. We made a soluble form with a tag on it. And we took human cross-section of bronchus. And we put it on there and said, well, where's the ligand? We know something's got to be binding. And we looked at it in Sialic 8 and Sialic 9. So here is Sialic 8, our construct, looking for its glycan ligand. And where does it show up? This is H&E cross stain. The red here in the submucosal glands. So our submucosal glands are generating something to engage Sialic 8. By comparison, this is the same with Sialic 9. It's almost the same molecule. It's a completely different pattern. Yes, it's in the submucosal glands, but it's out here on the epithelium. That's kind of where we'd expect it to be if it's going to be engaging these cells that are causing disease. And so we looked at this and we said, what's going on here? It's got to be out there somewhere. And we realized, of course, that we're working with postmortem tissue. These are airways that have been well flushed. And if we look at the submucosal glands, we see, and this is H&E, and this is stained for Sialic 8 ligand, that is putting the Sialic 8 on there to find its sugars. It's in the submucosal glands, and it's in the ducts. These are ducts here, and you've got guck and getting pushed out. Well, where does that go? That goes out to the surface, right? So we're figuring, oh, this must be a soluble molecule that's on the surface. Here's our post-mortem human airway. This is what it looks like. But how are we going to study this? And one of the advantages here, by the way, is that you have lots of colleagues in different areas that you can reach out to, and they are working with you to find this. So we have a great colleague at the sinus clinic, runs the Jean Kim, she runs the sinus clinic. And every time she does a sinus surgery, the first thing they do on these airways is pump in a bunch of saline and then have the person lean over and it all drains out. That's how they wash away the surface layer before surgery. They gave it to us. We have it. So we looked at a whole bunch of patients by taking that gunk, running it on gels, and overlaying, again, with Sialic 8. And what did we expect? Well, it's binding to sialic acids. Aren't there 100 or 1,000 sialoglycans out there? There might be, but it bound to one thing in human mucus layer airways. And it's about 1 million molecular weight. Yes, these are very special gels. And you see it in differential amounts in every flushing. You can imagine these are humans and it's different, but I'll show you some data on that. So it's a single major species, about 1 million molecular weight in every donor, and the levels were highly variable, donor to donor. So one of the first things we did is ask, well, is that binding to a sialylated keratin sulfate? We could ask that because we had the assay. We could just take that gunk we got and treat it with sialidase to knock off the sialic acid, treat it with keratinase, which is very specific for cutting these characteristic chains. And when we did that, so this is controls, and this is binding to samples that were treated with keratinase or sialidase, you can see we knock it out. So now we know there's 1 million molecular weight protein out on our airways that's carrying these sialylated keratin sulfates that are engaging Sialic 8. Because we could purify it, and we purified it by affinity chromatography using Sialic 8 as the trap, we pulled it down and we did proteomics, and it turns out to be a single protein called DMBT1. Don't ask me why. This is a common surface protein on, it's also called GP340 and has other names, that is on mucous airways. And in upper airways, we found a whole bunch of peptides. Those are all the green sections were identified by proteomic mass spec. But interestingly, this molecule has these intervening sections, which were not found, of course, because they're heavily glycosylated, and proteomic mass spec misses those. So this is a heavily glycosylated molecule. So we then stained for Sialic F and DMBT1 and they exactly overlay. And we see in submucosal glands, they're made again, just like that, in the same submucosal glands and cranked out. It varies. This is overlays. This is Sialic 8 ligand, DMBT1. And you can see it overlays exactly in every patient, but different amounts. And if we look at how heavily represented these Sialic 8 interacting DMBT1s, and it's not much, it's a subpopulation, they are more highly represented in patients that have inflammatory disease, chronic rhinosinusitis with polyposis. So here's our model. Nascent DMBT1 moves across the Golgi apparatus. There are enzymes in there that do or don't. So this is always heavily glycosylated, but sometimes they just don't make that target for Sialic 8. Sometimes they do. In these serosal cells, they get pumped out onto the surface. And if you have this, and you make more of it if you are inflamed, then you're going to turn off the eosinophils and mast cells. Because of the hour, and that is actually correct, I'm actually going to stop here. I will briefly say, sorry, I will briefly say that Sialics are now targets in Alzheimer's disease. That's because in genome-wide association studies, one of the Sialics, Sialic 3, showed up consistently as a molecule that predicts susceptibility to AD. If you overexpress Sialic 3, you're more susceptible to AD. If you express rarely a Sialic 3 that doesn't bind sialic acid, you're protected. What's going on? Well, we know these are inhibitory. They inhibit your microglia. If you have too much inhibitory signal, your microglia can't clean up the trash. And we went into the brain and found the molecule there that's involved. And the short story is it's 1 million molecular weight. It's a sialylated keratin sulfate. It's a single molecule. It's not DMBT1. It's a different carrier, but it doesn't matter. The Golgi apparatus is designed to take a molecule that's convenient in that tissue and decorate it very specifically with a signal that says we are going to downregulate inflammatory responses in this tissue. So now it becomes a target for modulating microglial phagocytosis in the brain. Maybe there's a sugar we could use to block that interaction. We free up microglia, they can now do their business of cleaning out the trash. You're less susceptible to Alzheimer's disease. Complex, yes, but now on the radar. I'm going to stop there and leave a full 4 minutes, because if anybody has any questions, I'd love to hear them. Please don't be shy. And I'm happy to reflect on the field or on our work and give you feedback. So.
Question from the audience?
Thank you. That was fantastic talk. Wanted to pick up on something you said about, you didn't say it, but you may have implied, or at least that's how I interpret it, is that our typical experimental models for mice and other good model systems to replicate like a biology that you see in humans. And say that because there's several food molecules, for example, that have different, the number of overlapping asparagines where you have ending glycosylations vastly different across those subset of proteins.
The answer is that you have to test it in each case. So we have evolved quickly, especially in certain areas. So I did show you data from the selectins that the mouse copies the human. E and P selectin are responsible for neutrophil extravasation in the mouse. They're responsible for neutrophil extravasation in humans, and the same structures involved. However, Sialics, which are immune inhibitory, you could imagine a pathogen would love that. What if I can just turn off the immune system? And in that field, things have rapidly changed, and animal models aren't perfect. Now, I will share that and this surprised us, delightfully surprised us. My graduate student in tech were doing this experiment, and I said, that's a stupid experiment. And they were right, despite my particular point of view. And that in what area of the human body wouldn't be susceptible to that kind of evolutionary pressure in pathogen immune system? Well, how about the brain? It turns out that our Sialics in the brain and their functions are copied between human and mouse. So in each case, you have to kind of probe that and discover that. But the bottom line is, especially in Sialics as therapeutics, you want to use a mouse model, you have to make a humanized mouse.
So you talked a lot about the role of changing the decoration of these extracellular proteoglycans. And we know if you change the expression of these glycosyl-transferases, obviously that can change the decoration. Can you comment on how the cell may control where in the apparatus these enzymes are being located and how that may change glycosylation?
So that's a very astute question. Thank you. We don't know enough about the suborganelle organization in the Golgi apparatus. We know these enzymes are in the Golgi. We know, for example, so because the brain is a immune protected space, we were able to recapitulate our studies on the microglial Sialic 8 target in mice. When we knock out the carrier protein, DMBT1, the enzymes that are there do not put that decoration on anything else. It drives me crazy. I can't figure that out. As you say, all the enzymes are there. So what am I thinking? I mean, is it that there are sub-directions? It's like you have to go down this street to get this kind of car wash. So it's like, how's that happening? And the answer is, we haven't the slightest idea. And of course, we've looked for expression of these genes in GWAS databases, and we don't see big changes in GWAS databases. So we don't know how this is being regulated.
Okay, last question.
So what regulates expression of VMPT1? Proteins that get decorated?
Again, that's something I don't have a sufficient background on to. So but frankly, and the reason I've kind of not focused on that is because when we look at DMBT1 in the human brain, it's got 5 isotypes. Only the largest isotype gets decorated, and only a portion of the largest isotype gets decorated. I don't think that these signals are being driven by expression of the carrier proteins. I think they're being driven, as was suggested, by maybe where, how much, and when the decorating enzymes, which are glycosyltransferases and carbohydrate sulfotransferases, are expressed. That's our challenge. Thanks.
Sorry, we didn't check online if there are any questions because we're already passing the hour. Please join me again to thank, Dr.